Hello everyone, my name is Lylla Almosd and I'm a master's student studying microbial biology at Rutgers University, New Brunswick. And today I will be presenting this research project that I've been doing for my thesis. So first let's talk about antibiotic resistance. Antibiotic resistance is one of the most urgent global health threats. So each year there's an estimated 7.7 million deaths associated with bacterial infections. And of that, 1.27 million deaths are caused specifically by bacterial pathogens resistant to antibiotics. And nearly 5 million deaths are due to bacteria associated with antibiotic resistance. And this threat has been increasing in severity, especially after the recent COVID pandemic, and it has been projected to keep increasing over time. Agricultural environments in particular have, especially livestock production systems, have become hotspots of antibiotic resistance due to the widespread use of antibiotics for growth promotion and disease prevention in their animals. Now, while many studies have focused on antibiotic resistance in the manure or the soils in these environments, less is known about the air, which is arguably more of a concern in relation to human health. Another problem we wanted to explore in our research is MRSA in farm environments. So why are we looking at MRSA? Well, studies have found an epidemiological link between MRSA infections and agricultural settings. This study shown in this slide has found that the closer you live to a site with manure, the more likely you are to get a MRSA infection. Now, this is alarming because according to the CDC, there's an estimated over 300,000 cases of MRSA. And this is concerning because although there are some treatments available, MRSA has become resistant to many of the first-line antibiotics that we use to treat it. And if left untreated, it can cause sepsis and even death. Therefore, we were interested to see if MRSA could be in the air at these local farm environments. And when we talk about sampling the air, we're talking specifically about sampling the bioaerosols. So these are suspended biological particles in the atmosphere that comprise of bacteria, fungi, microbes, and other non-living components like animal or plant debris. They make up a significant fraction of respirable particulate matter, PM2.5, which it refers to particulate matter smaller than 2.5 micrometers. Now, PM2.5 is particularly important because studies have shown that these PM2.5 can act as a carrier for bacteria and potentially pathogens as well. And this is dangerous because PM2.5 can penetrate deep into the respiratory tract since it is so small and therefore potentially transfer these bacteria and resistance genes to humans and animals. So these implications for human health are why we wanted to include it in our study. So our objectives were to quantify the cultivable heterotrophic bacteria and MRSA, as well as fecal microbes in bioaerosols and manure, and quantify the antibiotic resistance genes and total DNA extracts from these matrices. And we also will, in the future, do community analysis on our samples. So how did we do this? We utilized two different air samplers. So on the top left, you can see what we refer to as the active sampler, which is basically using PTFE filters connected to a vacuum pump that pulls the air through the filter to collect the bioaerosols onto the filter. And we set this for 4 hours at 30 liters per minute, collecting 7.2 meters cubed of air. And we also used a passive sampler, so specifically the Rutgers electrostatic passive sampler, which was set up for 1 week at a time in parallel to the active samplers. Now, with these bioaerosols, we plated them to quantify the heterotrophs, detect MRSA and fecal microbes as well, and with separate aliquots of the eluate, we filter concentrated it and extracted the DNA and did qPCR for SUL1 and 16S rRNA. SUL1 is a common resistance gene that people use to measure sulfonamide resistance, and it's widespread in the environment, so that's why we wanted to choose that one in particular. And in the future, we will definitely be doing qPCR for more antibiotic resistance genes as well, and also doing amplicon sequencing as well. So here are some pictures to help you visualize what our setup looked like. So we put the active and passive samplers at both ends of the cow barn here. You can see I circled the passive sampler hanging up nearby to the active sampler. And here's what it looked like in the pig barns. And here's the passive sampler hanging up nearby. So also we sampled the air at a control site at a nearby field, just as a comparison. Now on to our results. So we did not detect MRSA in the cow barn using the active samplers on any of the dates, but we did detect it at the pig barn at 2 out of the 5 dates. For the passive samplers, we detected it in both the cow and the pig barns on 2 out of 6 dates, and in the manure, we detected MRSA in 3 out of 5 of the dates, and the control samples, we did not detect MRSA, which was the positive result for MRSA is indicated by a pink or mauve colony on the plates. Now onto cultivation data. So here you can see the colony forming units for the active samplers and the passive samplers on the right, put on log scale for easy visualization. But as you can see, the pig samples had a higher CFU counts compared to the cow samples, and the cow had higher CFU counts compared to the control samples for both the active and the passive samplers. Now, this indicates, it suggests that bioaerosols from the pig barns may have higher bacterial loads compared to those in the cow barns when captured by the active samplers. And we also found a strong positive correlation between the CFU per meters cubed of the active samplers at the cow and pig barns compared to the average PM2.5 taken during the period of active sampling. And finally, for cultivation, we have the manure data. So manure showed a high bacterial load as we expected. Now onto qPCR data. First, we will be looking at 16S rRNA gene abundance. So as you can see, the control yielded the lowest amount of gene copies per meters cubed, but it was a bit similar to that of the cow samples, and pig samples had comparatively higher 16S gene copies. Now, looking at SOL1, you can see a similar trend where the cow and the control samples have similar gene abundance in SOL1, whereas the pig had a higher abundance of SOL1. And here's that same data for the passive samplers. Looking at 16S, you can see that the pig had higher 16S rRNA gene copies than the cow. And looking at SOL1, we can see that similar trend. Just to note here, we will be doing the control samples as well. This is just really early in our study, so yes. Next, continuing on to the qPCR data, we displayed the normalized SOL1 abundance by sampler type. And when I say normalized, I mean we divided the SOL1 copies by the 16S gene copies to reflect the relative abundance of SOL1 in our samples. So the proportion of bacteria carrying SOL1 independent of total bacterial counts. So as you can see, the relative abundance of SOL1 in the control and the cow sample were pretty similar in the active samplers, and a little bit similar to the pig as well, since that box plot kind of overlaps with that of the control samples. And when utilizing the passive samplers, we see similar results in that the cow and the pig have similar normalized SOL1 data. So these normalized values appear to be more similar across samples. Therefore, that trend we were seeing of higher gene abundance and bacterial counts in pig is no longer maintained once we normalize our data to 16S. However, in the manure, we have a more negative normalization value, which is likely indicative of the high concentration of total bacteria in the manure. So to end, in conclusion, we detected MRSA on some dates for the cow and the pig barns, and some of the manure samples, but in none of the background samples. Cultivable heterotrophic bacteria were present in bioaerosols, higher in pig than in cow, and higher in cow than in background, with both sampler types. And heterotrophic CFU per meters cubed correlated with PM2.5 for the cow and the pig active samplers. And also, the SOL1, the gene for sulfonamide resistance, was detected in bioaerosols from all of these sites, being the highest in the pig and comparable between the cow and the background bioaerosol samples. And these findings show that both cultivable bacteria and resistance genes are present in the air, suggesting antibiotic resistance may spread through airborne pathways in addition to those direct contacts. And these results highlight a potential occupational and environmental health hazard. And finally, thank you to everyone who helped out with this research and the NSF and Rutgers LSA.
Thank you. Questions?
Very interesting preliminary results. I wonder, could you isolate the Staph aureus from the aerosol cultivation, whatever, passive, I don't know what to call them, you know what I mean? But
Yeah.
from the air. And then actually then score them to see if they're MRSA strains. But even more importantly, do some sequencing on them and find out what their clonal complex is, and to see if they're actually of human origin or of animal origin, and if you can maybe trace, like maybe there's one student that works at the farm that's a MRSA carrier, right? And you're just picking up, every day that guy shows up or woman shows up, those are the bacteria that you're finding on the filters or something along those lines. Does that make sense?
Yeah, that's very interesting. We haven't gotten that far yet, but that is definitely something we would consider doing.
Any other questions? Okay. Let's move on to the